A very good afternoon, everyone.
Am I audible?
We are about to start scientific session in next 5 minutes. Modern college of Pharmacy Nigdi is a most reputed institution located in a prime area of Nigdi, Pune since 1998. It is a part of most prestigious progressive education society that is imparting education since 1935. The college is permanently affiliated to Savitribai Phule Pune University. The name of society and so the college typifies the society's mission to be progressive in education and modern in technical knowledge. The college is approved by Government of Maharashtra AICTE, recognized by PCI and approved under section 2F and 12B of UGC Act 1956. Under the leadership of Principal Dr. P.D. Chaudhary, the institute is running basic undergraduate course in pharmacy, B.Pharm, 4 postgraduate courses in pharmacy that are M.Pharm in pharmaceutics, pharmaceutical chemistry, pharmacology and pharmaceutical quality assurance. Also, successfully running PharmD and PharmD post-baccalaureate courses. Institution is a recognized research center for PhD by Savitribai Phule Pune University with 17 faculty as research guides, and under their mentorship, total 30 research scholars have successfully completed their PhD. As a golden feather in the crown of modern college, it has been honored with Best College Award by Savitribai Phule Pune University. It is a matter of pride that institution has been accredited by National Assessment Accreditation Council Committee with A+ grade with 3.47 CGPA. Also recognized by National Board of Accreditation and is proudly securing rank in under top 100 institutions all over India as per National Institutional Ranking Framework since last 6 years, and has been honored with platinum grade in AICTE CII survey for 3 consecutive years. Research ambience is the distinctiveness of the modern college. Institute has various sophisticated research equipments worth more than rupees 3 crores. As a consequence, faculty has been engaged in 149 IPR activities and has published 667 articles in various national international journals in last 10 years. Faculty has published 142 conference proceedings, 89 books and book chapters. Numerous faculty have been honored with various research awards like Women Researcher Award from VD Good, Dr. R.L. Nikor Award, IDMA Award, Devang Mehta Educational Award and Best Research Work Award. Institute has received grants from various government and non-governmental agencies like UGC, IBRO, AICTE, BCUD, DST for research projects. Endowments worth rupees 2.35 crores. Total 17 faculties are active editors and reviewers for national and international journals. Many of the college faculty are actively engaged in offering research consultancy and have garnered more than 4 lakhs through 3 consultancy projects. In collaboration with Grinson Pharmaceuticals, the faculty have developed and marketed 2 pharmaceutical formulations, MCOP's Dibenyl capsules and MCOP's Health Plus tonic. Every year, Modern College organizes various guest lectures, seminars, workshops and conferences sponsored by AICTE, ICMR, SPPU and many more, and has more than 40 academic and industrial research partners. Holistic development of students is the best practice followed by institute. Along with research quotient, students are also promoted to take active participation in social activities like health checkup camp, cleaning campaign and many more. Institutional NSS unit has been rewarded as Best NSS Unit by SPPU, and faculty has been rewarded as Best NSS Officers at university level. Students shine like stars in the universe of academics. More than 300 students are university rankers and 5 are gold medalists. Most of the passed out students opt for higher education and many has been placed in various national international firms through institutional training and placement cell. Potential fresher students get opportunities with highest package of 8 lakhs per annum. The stalwarts produced by the institute are playing a vital role in the society as global head clinical trial, vice president, DGM, global PV auditor, global manager, senior research scientist, managers, entrepreneurs, principal project manager, and many more. All embracing, it could be said that Progressive Education Society's Modern College of Pharmacy Nigdi is exploring every avenue open to it, and the winning strike continues.
A very good afternoon, everyone. I, Mrs. Rupali Jinturkar, would like to welcome you all to the day 3 of Indo-US International Conference entitled Unlocking Horizons in Global Multidisciplinary Healthcare Research, organized by Progressive Education Society's Modern College of Pharmacy Nigdi Pune in collaboration with the Association of Pharmacy Professionals. I extend a very warm welcome to everyone here on behalf of convener of the conference, Dr. P.D. Chaudhary, Principal, PES Modern College of Pharmacy, Nigdi, Pune. Dear delegates, on day 1 of the conference, we had inaugural function graced by Chief Guest Dr. Shamakant Shimpi and Chief Patron Honorable Dr. G.R. Egbote sir, followed by 2 scientific sessions by Dr. Javed Ali and Dr. Vijay Masan. Later, we had some poster and oral presentations, and the day 1 concluded with a spectacular cultural event. Day 2 started with keynote addresses by APP senior executives, later 2 scientific talks by APP associates, followed by scientific presentations. Today, the day 3 of conference, all the presentation sessions were completed and now we have 5th scientific session of the conference by Dr. Arabinda Ghosh, Department of Molecular Biology and Bioinformatics, Tripura University. He will be enlightening us with a topic, the era of modern drug discovery, unveiling the transformative potential of in silico methodologies. I welcome him on behalf of convener and principal, Dr. P.D. Chaudhary, sir. Welcome you, sir. I also welcome and extend my warm greetings to Chairman of the session, Dr. Sunil Jalalpure, Principal and Professor, Department of Pharmacognosy, KLE College of Pharmacy, Belgavi, Karnataka, and Co-Chairman of the session, Dr. Parasuraman P., Department of Pharmaceutical Chemistry, MS Ramai University of Applied Sciences, Bengaluru. Now, I would like to call upon Dr. Somdatta Chaudhary, Associate Professor, Pharmaceutical Chemistry Department, to give introduction of speaker, chairman, and co-chairman of the session. Over to you, Somdatta sir.
Thank you so much, madam. Am I audible?
Yes, sir.
So, ladies and gentlemen, first of all, welcome to all of you for the last session of this conference. As we gather here in this meeting, I'm reminded of an incredible power of a collective curiosity and the pursuit of knowledge. We are ready to embark on an hour of insightful presentation and stimulating discussions. So today we have a Dr. Arabinda, a distinguished speaker who will share their latest research and insight pushing the boundaries of a era of a modern drug discovery, unveiling transformative potential of in silico methodologies. I encourage each of you to engage actively, ask questions, share your ideas. Let's make this interactive session as fruitful as possible. So, with this, we have Dr. Sunil Jalalpure as a chairman for this scientific session, Dr. Parshuraman Pavadai, co-chairman for this scientific session. So today we have it's my privilege to introduce a big stalwart in the field of pharmacognosy, then pharmacochemistry and bioinformatics, like Professor Sunil Jalalpure, Dr. Parshuraman, and Dr. Arabinda. So first, I want to introduce Dr. Jalalpure. Professor Jalalpure serves as a principal and professor at a KLE College of Pharmacy, Belgavi. A, a scholar of immense repute, Professor Jalalpure has cultivated a rich educational foundation with the B.Pharm from Karnataka University and advanced degrees including M.Pharm and PhD from Rajiv Gandhi University of Health Sciences. His expertise is backed by extensive research training at Rhodes University in South Africa. An acclaimed academic, Professor Jalalpure has been honored with the AICTE National Technical Teachers Award in the 2022 and recognized as a base scientist by the KLE Academy in 2020-21. He has many awards like this in his credits, but I mention only latest one for the time constraint. His contribution to research are substantial with over 161 scientific papers and 6 granted patents under his belt. Professor Jalalpure's research interests span from traditional pharmacognosy to the cutting-edge green nanotechnology and nanoparticle drug delivery system, positioning him at the forefront of innovative pharmaceutical sciences. As we proceed, let us draw inspiration from his exemplary career, dedicating ourselves to pursuit of knowledge and innovation in the immense humanity. He has a many more achievements to his credit, but unfortunately, they are too numerous to mention here. Apologize for any omissions. Please join me in welcoming Professor Dr. Sunil Jalalpure. Then Dr. Parshuraman Pavadai is a distinguished researcher and academician with extensive expertise in the computational and experimental pharmaceutical sciences, currently serving as an associate professor in the Department of Pharmaceutical Chemistry at Ramaiah University of Applied Sciences, Bangalore. Dr. Pavadai has built a remarkable career in the drug discovery and development. His research focuses on cutting-edge techniques such as computational drug design, QSAR, pharmacophore modeling, molecular dynamics simulation, and molecular docking. His work also extends to the organic synthesis, then DFT studies. Dr. Pavadai has a strong inclination toward interdisciplinary research, evident from his contribution to synthetic medicinal chemistry, phytochemical isolation, pharmacological screening of both natural as well as synthetic compounds. His research spans a variety of therapeutic areas, including cancer, Alzheimer, infectious diseases, and inflammatory disorder. With over 100 publications in his credit, which are indexed in the Scopus as well as SCIE, Dr. Pavadai has made a significant contribution to the pharmaceutical and medicinal chemistry. He has also authored several book chapters, then filed multiple patents related to the novel drug delivery systems and therapeutic compounds. In addition to his academic and research accomplishment, Dr. Pavadai has recognized as an effective mentor and educator. He has guided numerous B.Pharm, M.Pharm, and PhD students in his research project. His innovative teaching method and dedication to the student development has earned his accolades such as the annual exemplary faculty award, best research award, with a proven record of blending computational tools with the experimental methodologies. Dr. Pavadai continues to drive advancement in the drug discovery, nanotechnology, and targeted therapeutics which are contributing to the evolution of modern pharmaceutical sciences. Please join me to welcoming Dr. Pavadi. Then now, without further ado, let us welcome to our speaker, Dr. Arvind Ghosh, who will talk about the era of a modern drug discovery, unveiling the transformative potential of in silico methodologies. Dr. Arvinda stand as a prominent figure in the realm of biomedical sciences, characterized by his extensive expertise in the molecular biology, bioinformatics, and microbiology. His multifaceted career, marked by significant academic and research contribution, underscores his dedication to advancing scientific knowledge and its practical applications. Currently, Dr. Ghosh serves as an assistant professor at Tripura University, where he imparts his knowledge and guides next generation of scientists. His prior faculty position at Mahapurusha Srimanta Shankaradeva Vishwavidyalaya and Guwahati University has enriched his experience in a diverse academic settings. He earned his PhD in Biosciences and Bioengineering from prestigious Indian Institute of Technology, Guwahati, providing him with a robust foundation in the cutting-edge research methodologies. His technical proficiency encompasses a wide range of laboratory and computational techniques, including molecular technique, chromatography, spectroscopy, and computational biology. In the vast landscape of scientific endeavor, Dr. Ghosh work is in the biophysics, biomedical sciences, shines with the exceptional brilliance, earning him a place among the world's top 2% scientists as recognized by the Elsewhere and Stanford University 2024. Dr. Ghosh research endeavors are characterized by pro- prolific publication record with a substantial number of a high impact research paper, over 195 in the number, reflecting his commitment to the disseminating scientific findings. His innovative spirit in further evidenced by his securing multiple patents, which are near about 10, demonstrating his ability to translate research into the tangible outcomes. He has about he is also secured one project by Prime Minister, which demonstrate his ability to obtain competitive grant funding. It is near about 1 crore. His research interests lie at the forefront of biomedical sciences with a focus on computational drug discovery, utilizing computational tool to identify and develop novel therapeutic agent, cancer research, investigating a molecular mechanism of cancer and exploring potential treatment strategies, infectious diseases, contributing to the understanding and combating the infectious pathogens. Dr. Ghosh actively engages in a peer review, ensuring the quality of a rigor scientific publication. He also serves as a editorial board member of a various journal, including many Cuban journal, contributing to the disseminating with a high quality research. This is very important part of contributing to the scientific community. His work is published in many high impact factor journals. His participation is scientific in the scientific community activities shows his dedication to the advancement of science. In summary, Dr. Arvinda Ghosh career is a testament to his dedication to scientific excellence. His contribution to the research, education, and scientific community have made a significant impact in the field of biomedical sciences. Please give a warm round of applause for to welcome him. Really eager to hear this session as of big energies are coming together like pharmacognosy, in the field of pharmacognosy, pharmaceutical chemistry, and bioinformatics. Of course, I am talking about the network pharmacology, which is which is a demand of a today's researcher. And therefore, Dr. Arvinda is going to highlight this by his talk. Let us welcome him to talk on this. After his talk, Dr. Sunil Jalalpure and Dr. Pavadai will summarize the session. Thank you so much. And let's welcome Dr. Arvinda Ghosh. Welcome, sir.
Thank you, thank you. Thank you, Dr. Chaudhary, for introducing me in this session. And I'm really very much grateful that I got the invitation in such a big event to speak a small, there's few things about my research work and whatever I'm doing here. During this academic career, I have already changed three universities. This is my third university that I have joined, and really I have gone through but I have seen a lot of new things while changing the universities, new faces, new students, their ideas, thoughts, and really that helped me to learn a lot during this process. And definitely, I'm associated with Professor Chaudhary for a long period of time, and I have learned a lot from him also. So we had a very good connection regarding our research work also. So thank you very much, Dr. Chaudhary, for introducing me. And here I'm going to share my screen so that I can present my work. Let's
Sir, we have given a rights to you. You can share your screen.
screen. I think. So is it visible?
Yes, sir. Yes, it is visible. You can start.
So is my screen visible, right?
Yes, sir. Yes, sir. Yes, yes.
Okay, okay. Thank you very much. If at some point my voice is not audible or screen is not visible, please let me know. Otherwise, I'll keep on going with the flow. So that will be difficult for you to understand. But I'll try to give. This is a very basic thing I'm going to tell. That what modern drug discovery demands nowadays, and how actually we as a faculty, as a teacher, and as a researcher, we are trying to unveil the process to develop new or to find out a new drug so that that can be helpful to the society. I think everyone we know about the post-pandemic era of COVID. So after that, the drug discovery process has taken a huge role. And after that, there is a huge development of genomics and modern sequencing technologies. So therefore, when we have a big amount of data with us after the post-pandemic period, having lots of multiple parallel sequencing, high throughput sequencing technologies, we are having very huge amount of data with us, and with that we can play for the development of new drug or to design a new drug. So there are few terminologies, and you know better than me, as because you are all pharmacologists. Specifically, I'm just a biologist. So here I'd like to speak about the topic today: the era of modern drug discovery, unveiling the transformative potential of in silico methodologies. Here, actually, this entire presentation will tell you how to proceed through, like if some students are watching, they can observe how we are proceeding through the methods, okay, to publish a research work, what a research work demands nowadays, like what are the things we are going to give, what should be the output. So this presentation, I think that will be helpful for them. So here I'd like to tell a little bit about the introduction. So modern era, as I told, that post-pandemic period, when we have a high throughput sequencing data available with us, that will helpful, and it has transformed the landscape of pharmaceutical development, integrating advanced technologies, innovative methodologies, and a deeper understanding of biological systems. So nowadays, when we have a big data with us, exceptional computational science already integrated with biological science, pharmaceutical science. We are having like modern day AI methodologies, like we are having machine learning approaches. Like yesterday, I think whatever I have gone through the topic of Professor Masant, he has also informed about the AI methodologies, how to perform QSAR or IQSAR. So that has taken into course nowadays, and that is very helpful in order to get high throughput analysis within a very short period of time to get a large data set to resolve, and finally will come into a conclusion regarding a disease, regarding a particular medicine or a drug to apply on to patients. So what are the key components of modern drug discovery? First and foremost thing nowadays, a very important, like genomics and target identification. That means we have to understand the whole genome first. So we are having various technologies like Illumina or PacBio, different methods we can use depending on the size of the length of the whole genome. Okay, we can go for shotgun sequencing and various methods. So after having this data, sequencing data, we should go for the genome annotation. Just that is the very important part. So that needs lots of knowledge, that needs lots of statistics, good amount of mathematical idea also. So after having those information with us, that means if we have the proper annotation of the genes that are available within a particular genome, so that will helpful for us to determine a particular target. So for example, we are taking a patient sample. The patient is suffering from a particular cancer or suffering from particular viral infection. So from there, we can check the numbers of genes that are available within the whole genome, and the numbers of genes those are upregulating or downregulating. And if we have those kind of information with us, that will be helpful to identify the specific target on which we want to target our drug that we are going to design. Then when you have the genomics information and the target identification done, so we can go for high throughput screening of molecules. We can go for small molecules, we can go for peptides, we can do various combinatorial studies. So we can perform screening of such molecules which can have a potential to become a lead compound or can be a drug compound. So this will helpful for us when we perform the high throughput screening, and finally we'll end up into a small number of molecules through which we can identify whether which molecule will be better for the treatment of particular disease. So apart from that, there are few things that these are the two things that we are usually doing this regularly in our laboratories. Mostly people are depending on these two things. People are doing for the publication, for patenting, and many other things. But this is the era, again, this is the era of artificial intelligence and machine learning. This is very important. When artificial intelligence and machine learning has took the count, when they get into the account in biology, they actually reduce the large amount of time that we were wasting earlier while doing the experimental studies in the laboratory. So we can do to develop a drug, we can perform in computational science or in a computer, and after having the data with us, we can go for laboratory experiments. So when we integrate genomics, when we integrate high throughput analysis, artificial intelligence and machine learning, we can develop personalized medicine. This is a very pronounced nowadays, personalized medicine. So in person, we can go for, we can develop a drug for particular disease. Okay. But the biggest thing, that is the funding. When we'll have a ample amount of funding with us, then this kind of research can be carried out easily. Otherwise, it will be very difficult. Therefore, we have a solution. When we have lack of funding, like the country like India, so there are, we know that there's a large amount of population, large amount of faculties and scientists regularly they are applying for funding to Government of India. But Government of India or the other funding agencies, how many projects they will consider to release funds. So there are some limitations. When we are under certain limitation, then we should go for collaborative research. and open for innovation. So in this way, we can connect throughout the world. We can go for open collaborative research. The part that is possible in our laboratory that will perform, the part that is possible in other laboratories will combine the work, and finally we can get a conclusion. So accordingly, we can do a huge amount of work, and we can develop various new methodologies, techniques for modern drug discovery. So here is some workflow that I'm going to tell today. Like the first and foremost thing that we are looking is the gene expression and target identification. So for that, we'll be using some database for differential gene expression, and then we can perform some Venn analysis in order to get the common genes that are present within a treated drug, non-treated drug, or a disease patient, or a normal patient. Okay, then we can go for network and functional analysis. So that will in this study we'll be doing like construction of the protein-protein interaction network, identify hub genes, and will perform the functional enrichment to understand the better way about certain disease and the efficiency of the drug and the genes that are responsible within a particular pathway. Okay, then the third step is the drug discovery and validation. So in this step, we are following like ligand selection, high throughput virtual screening, DFT calculations, that density functional theory. Sometimes, you know, it is important, but sometimes we are not following that DFT. We are just bypassing DFT, and we are doing our research. That is essential. I'll tell why is it necessary during my lecture. Then redocking for validation. So validation is important when the molecular interaction of small molecule with the target protein or the target DNA or RNA, or a peptide molecule, when it is combined with a DNA RNA target or a protein target, it is very important to validate. Otherwise, we may give a wrong information to the society, and based on that, when you go for experimental studies, we develop a new drug or a peptide or some other type of molecules, and we give to the patient, like in the clinical trial, that fails. During the COVID period, approximately there are 10 millions of publications in 2020 to 22. So there are 10 million publications on the COVID, and we do not have a single drug available in the market for COVID. Many researches going on. We are having a vaccine, so that already most of us we have taken, but we do not have a small molecule drug available or some other functional drug available for COVID. It's not available, as because we know the nature of the virus. So it is very difficult. Although in pen and paper everything is available, but in reality that is not available. So there are some limitations. Then finally, in order to accomplish our task, we perform the molecular dynamic simulation to see whether the target and the molecule interaction is quite well stable or not with respect to time. So that is very important. So we'll be discussing. So first I'll start with the differentially expressed genes. So for these, in order to understand the genes that are expressed differentially between two or more conditions, such as disease versus control, treated or untreated, or different time points in an experiment. This is very important. So therefore, differential gene expressions are important to understand. Then several databases provide publicly available DG data from transcriptomic studies, including RNA-seq and microarray datasets. So if we go to the differentially expressed genes database, we can see this kind of information available with us. Like the most popular database is the Gene Expression Omnibus, this GEO. Apart from that, we are having Array Express and TCGA, the Cancer Genome Atlas. So from these databases, we can collect the data of differentially expressed genes of a particular disease, which is under various conditions, like the conditions that I have mentioned here. So what are the applications of DEG analysis? So it will identify cancer-related genes if we are working on cancer by comparing tumor and non-tumor tissue, then studying immune response after infection or vaccination, finding drug targets by analyzing gene expression changes post-treatment. So these are the application, or I think I should tell the pros of using the differentially expressed gene datasets. So actually here I have incorporated one of our work, but that has not yet completed, that is not yet published. So this is just going on. So one of my research scholar is doing this work. So here we are working with HMPV. This is I think we know about this as a very recent occurrence in India. Few cases have already been reported from India. So we have used the gene expression profile obtained from a data set which is called GSE 157240 from a GEO database from NCBI, and out of we got total 26 samples, blood samples that has been taken worldwide. It is not restricted to a particular site, where 13 HMPV infected and 13 are non-infected patient sample. And finally, we got few information. We can see there are numbers of differentially expressed genes that has been expressed. That means overexpressed. Some are down-regulated. Like here you see blue dots are down-regulated. That means the genes that are down-regulated during the HMPV infection, and red dots are up-regulated genes that are overexpressed during HMPV infection. And these are the gray are, they are non-significant. So we are calculating the level of significance at p-value 0.05, and we are calculating the numbers of genes that are overexpressing. That is our interest now. But we can see the numbers of overexpressed genes are less as compared to the number of down-regulated genes. So we are working in this field now, and later on we'll go to this. So this work is still is incomplete. So therefore, I do not would like to say more about it. But I have just show the use of DEG, how we can use in our data set preparation. Then we can use the Venn diagram for target identification. So Venn diagrams are commonly used to compare multiple DEG datasets from different conditions. For example, disease versus control, drug treated and untreated, and identify common or unique genes in each dataset. So for example, we are having three datasets from patients, and if we want to see, like, one dataset is for drug treated, another dataset is not treated, another dataset normal human being. Now I want to see the common genes that are regulated, overexpressed, expressed, or down regulated during the process. So for example, like in case of machine learning, people are using clustering. So here we can use the Venn application, and we can identify the common and unique genes in each dataset, which can reveal potential biomarkers and therapeutic targets. So the tools that can be used for Venn diagram preparation, having the dataset with you, like BioVenn, Interactive Venn, and Venni 2.1. So these Venn diagrams showing the overlap of 40 differentially expressed genes among dataset 1 and 2 and 3, highlighting potential common biomarkers and therapeutic targets. We can see here from 3 different datasets, we are having the 40 number of common genes from the 3 datasets. So if we learn more about those 40 genes, then we can identify the prominent genes that are involved in that particular disease. Okay. So followed by this, we go for network pharmacology. Network pharmacology is a system biology approach that integrates computational and experimental techniques to study drug target interaction within complex biological networks. So that is very important. Now, what is this? Actually, very simply, if I would like to say that when we know about biochemical pathways, a biochemical pathway is being controlled by many numbers of genes and proteins. Pathway is controlled by many numbers of genes and proteins. Like in case of biochemical pathways, we are having enzymes, various enzymes. Like for example, glycolysis. There are several enzymes that are involved in the process of respiration and the glycolysis process, right? So in order to understand the interaction between these proteins, how they are regulating a biochemical pathway, how this is behaving during the process of a disease development. So the networking study is very important. So therefore, network pharmacology will give us that idea that how a biochemical pathway has been regulated under the various genes and proteins. It focuses on understanding the relationship between drugs, targets, and disease-related pathways rather than single target approach. So that is the important thing. Like earlier, prior to network pharmacology study, people used to do by studying literature, they go for single target analysis. Like even I did earlier when we started this working in this field. So we used to study many literature. From there, we used to find the target, the people there or the authors they have used in their study, and we have taken that target and finally you go for further analysis. But that is a limited work. Understanding a single target is a limited work, as because a particular type of biochemical event is not regulated by a single target. So it has been regulated by numbers of targets. So therefore, it is very important to understand network pharmacology nowadays. Otherwise, we will end up in somewhere which will not lead into production of any kind of productive drug or marketable drug. Okay. So it identifies potential drug targets through a systematic approach. So what are the key steps? The key steps in network pharmacology are protein-protein interaction, PPI network analysis, then identifying hub genes, functional enrichment analysis, cake pathway analysis. So these are the few key steps that we are following in network pharmacology. The tools that are used in network pharmacology. Why I have incorporated the information about tools, as because my topic is specifically on the methodologies. That's why I have been incorporated. This is very important. You know, I have attended many lectures from many other countries, even in the YouTubes that are available. But the many people, the lectures are there. They are not informing about the tools, which tool is used for particular kind of work. That's why we are always within a black box. So we have to use a lot of time, or you have to waste a lot of time to understand the tools or the methodologies, how to perform a particular work. So for the young generations or the young scientists, those who are going to work in this field later on, they can have some idea. So how to proceed. So like for network, in this network pharmacology, for PPI network analysis, we are using string database. We know about this string database for PPI interaction studies. There is another database which is called as string viruses. String database doesn't contain information about viruses, but nowadays string viruses are also available. There is another database which is called stitch database. So many databases available. So I have incorporated very few, as because we are working in this with these databases. Now then we can use Cytoscape. Cytoscape is an open source tool we can use for PPI network analysis. Then for the functional enrichment, we can use the database which is called as Debit. Okay. Then we can use ShinyGo and Gestalt. So we can use this kind of database servers. Then for the pathway enrichment analysis, we are using the cake database. That is, we know about this Kyoto Encyclopedia of Genes and Genomes. It's a very common, this is the best biochemical pathway database available at this moment. Now let's come to the string for PPI network construction. String, search tool for retrieval of interacting genes and proteins. The full form of string is search tool for retrieval of interacting genes and proteins, a database that provides known and predicted protein interaction. Most of us, we know about what is this. This is a network. For example, here is my gene of interest. Here is the protein of my interest, and how this protein is interacting to nearby proteins to regulate a particular biochemical pathway. Here, each and every protein, it is showing in the form of a node. The spherical round shape is a node, and the lines, these are edges. So the lines are telling what is the total confidence score. That means what is the relation between these protein with this protein. So how strongly they are interrelated. how strongly they are interrelated. These lines are called as edges. So number edges will tell the confidence, total confidence. Here we can see the highest confidence score, always we consider for that is 0.9. High confidence 0.7 is good, medium confidence 0.4, and low confidence 0.5. So when the confidence level is low, let's see, here is a single line. It has less confidence score. That means the interaction between two proteins may be taken place or will be taken place at certain conditions. So after the network analysis from the string database, string is providing a network which is based on our confidence score. Similarly, we can take this into the Cytoscape, which is an open source software for network visualization and analysis. Here you can see we have studied a particular we are working with a particular disease, and you can see the interactions of the numbers of genes that are interrelated within a pathway. Okay, so this is used for analyzing protein-protein interaction. That means Cytoscape can be used for protein-protein analysis interaction, gene network, and many more other studies. So what are the customized properties that we are using in Cytoscape? We can adjust node, edge color, layout for clarity. We can apply layouts, choose an appropriate layout like organic, circular, hierarchical, different types of layouts we can put. Here I'm showing you here is a circular layout, and we can also have the facility for export the network. And finally, we can export this entire network study. We can go for publication. Very good quality pictures come from this Cytoscape. From there, we can directly go for publication. Many people, apart from using this network analysis these tools, people are using R programming. From there also, you can perform this network analysis. Then the next step is, after the interaction study, what is our motto? Our motto is the principle to find out the principle or the major genes or the proteins that are involving in the disease that has the highest value or the highest importance within particular biochemical pathway. In order to understand that, we have to find out the hub genes. So there is a plugin called CytoHubba within the Cytoscape that is not available in the string database. So that is available in the Cytoscape. So this way Cytoscape is helpful to us in order to give us this kind of information. Okay, to give us the most probable or the best possible combination of the targets which are responsible in a particular disease. So the CytoHubba is a Cytoscape plugin used to identify hub genes in a network. Hub genes are key genes in a network within many connections to other genes. They are important because they control biological processes and disease mechanisms. So CytoHubba provides multiple methods to identify hub genes in a biological network. First one is maximal click centrality, degree, closeness, betweenness, etc. So these are various methods that we can use within the CytoHubba to better understand the genes. And here we have we have marked it red. That is the most important gene that is involved in that particular biochemical pathway in order to maintain a particular disease. Then functional enrichment analysis. The next step is functional analysis. So this method is used to understand biological roles of genes, proteins by grouping them into known functions, pathways, and processes. So earlier what we have done, we have just identified the genes. But the understanding of these genes within the pathway is yet to be done. So functional enrichment will help us to understand that. We have just determined the genes that are involved in a particular pathway. There are major genes that are involved in particular pathway to control or regulate a biochemical pathway or to regulate a disease. That was done till earlier part. In the functional enrichment analysis, we are going to determine the known functions of genes by grouping them to understand their pathways and processes. So there are a few analysis that we perform that is called as type of functional enrichment analysis that we have been jotted it out. Like first one is gene ontology analysis. Here we can study about the molecular function, what the gene does, for example, enzyme activity, biological process, the pathway it contributes to, like for example, apoptosis, or cellular component where it is located, mitochondria. You know, some proteins, or maybe you can see it has been located in the cytoplasm. Some proteins are located in the cell membrane. So how will you determine that? So that will be determined when we study the gene ontology analysis or enrichment analysis. Next one is cake pathway analysis. So by understanding this, we can understand the holistic way of a gene that is involved within a particular pathway in order to regulate a particular disease or a particular biological function. Next is how we will perform the gene ontology enrichment. Like just I told you about the gene ontology analysis, these few basic analysis we perform, but how we'll perform this enrichment analysis? So what are the key parameters that should keep in our mind? That is the FDR value, 4 enrichment value, and the P value. We have to calculate these values from the gene ontology study, and from there we can determine these values, and based on these values we can tell the involvement of a particular gene or a particular protein within a biochemical pathway and how they are important. So here are few diagrams. There are bar plots that has been incorporated. Like see, gene ontology enrichment analysis. It is a biological process include in response to biotic stimulus. Like here also we are studying one. We are doing one work. Our study is about the quorum sensing inhibition study. So we have identified some quorum sensing genes and their responsibilities in a particular biochemical pathway in biofilm formation, and we can see the response to biotic stimulus and various for enrichment analysis within various components, like these few components, like we have molecular function, biological process, and cellular component. Here we have performed, this is the molecular function. Then we can have the next function like trans-esterification activity, acetyl transferase activity, hexosyl transferase activity, various other activities that we can determine, like likewise the cellular function. Then where that particular protein will be located, that will be located in an extracellular region. So from there also, from the gene ontology enrichment analysis, we can identify in the cellular component where that particular protein will be located. So likewise, we can determine many functions of a particular gene and the protein that are involved within a biochemical pathway. Then finally we'll come to the cake pathway analysis. So cake pathway analysis is a bioinformatics approach used to understand the functional significance of genes and proteins by mapping them into biological pathways stored in Kyoto Encyclopedia of Genes and Genomes, the cake database. So here our study is going on about the quorum sensing, and we have determined few pathway interaction, how this has been involved. Like we can see quorum sensing, that particular gene that we are studying, that mostly involving quorum sensing, and it also involved in the biofilm formation in Pseudomonas aeruginosa. This is a common gene that is present in Pseudomonas aeruginosa and few other bacterial species that we have already studied from the cake pathway enrichment analysis. Once our gene or the target is determined, our gene and target, or gene or a protein target is already determined, the next step is for the drug development is the retrieval of the ligand structures. So we can retrieve the molecules from various databases. We can synthesize the molecule, then we can get the structure by using NMR, FTIR, different other methods. When we have the structures with us, we can have the ligand structures available for further studies. So there are several publicly accessible ligand databases that will help us on small molecules, bioactive compounds, and drug-like molecules. The databases are widely used in drug discovery, molecular docking, and pharmacological researches are, for example, Drug Bank, NPDB, PubChem, Zinc, Impaired database. There are many. Actually, there are numerous data, like food database, different databases. I have jotted down few. People most pronouncedly use these databases for their drug discovery work. Then once we have ligands, these databases will give us numerous numbers of data, like 1 lakh data, 1 lakh molecules, 2 lakh molecules, 50 lakhs molecules, like lakhs of molecules they will give. But which out of the lakhs of molecules, which one is better one for the treatment of the particular disease that we are looking for? How will you determine? For that, we have to go for virtual screening. That means we have to do some screening of the molecules, which one will be better. So for that, we are using some ligand selection criteria out of the numbers. This is the first step of our virtual screening, like we said HTBS. In the HTBS, mostly when we say HTBS, we think about the, like, either we go for QSAR or we can go for the like dock-based analysis. So docking-based analysis, like we perform Autodoc Vena to do this HTBS. But before performing HTBS, blindly 1 lakh molecule, it is not necessary. It is better to remove the unwanted molecules that are not required in our research. So we should have some selection criteria. So the few criterias that we have set for understanding the molecular properties. So like drug likeliness, synthesizing accessibility, ADME properties, log-P, lipophilicity, solubility, and toxicity. These are the few criteria. Based on these few criterias, we are considering few parameters, and after considering these parameters, we are thoroughly screening the molecules from 1 lakh to few hundreds of molecules. We are reducing the numbers, as because the numbers of molecules, they fail to pass these screening filters. When they fail to pass the screening filter, we are removing them, and whatever the molecules they pass through the screening filter, we keep them for further analysis. Then we go for we can perform ADME, we can perform pharmacochemical, then we can perform the pharmacokinetics. This is a very known data tool about Swiss ADME people are using. Apart from that, there are various other databases. Here I've included few. Sorry, here I have included few name like Swiss ADME, PKCSM. For toxicity, we can use ToxTree, we can use QuickProp, we can use the Protox. So various types of databases we can use for our study. So after performing ADME, we can shortlist few molecules, like I told you, like approximately from 1 lakh molecule to few hundreds of molecules. After having these hundreds of molecules, we can go for dock-based screening, or we can go for QSAR-based screening, and that will give us a very comprehensive information, rather than going for a very rigorous huge number of analysis, and it will take a lot of computational power when you go for 1 lakh molecule to analyze or go for high throughput screening without prior screening into like the criteria that I have mentioned here. Once the target protein is selected, the next step is to retrieve or predict the 3D structure. Sometimes when you predict the gene, when you predict the protein, we may not have the structure of the protein available in the protein database. That means three-dimensional data may not be available. If it is not available, at that time, what we have to do, we should go for the modeling of the protein. So we'll be using various tools to model the protein depending on the type of the protein that we are looking for. So for the structure-based drug design, that is a very core step, important step, that we should have a three-dimensional target structure available with us. Proper preparation of the target protein ensures accurate molecular docking and rest molecular dynamic simulation analysis. So for this, we should have a PDB structure available, retrieve the PDB structure, then removal of water molecules, and the associated ligands or the ion molecules which are present in the protein. We have to clean the protein first. Then we should determine the active site of the protein, and finally we should have hydrogen ion charges, and next study will be our docking. Or if the PDB structure is unavailable, what to do? Then at that time, we have to go for homology modeling or AI-based structure modeling, like we are using nowadays. We are using AlphaFold 3. 2 is already over. AlphaFold 2 is not available nowadays. AlphaFold 3 is available. Although if you type in Google AlphaFold 2, it will show, but it is not functioning. Okay, it may give you error information. So AlphaFold 3, they have refined. DeepMind's AlphaFold. So this year, people, the DeepMind's developer, he got the Nobel Prize for development of this AlphaFold, right? Then validation of the predicted protein structure is very important. So for that, we are using various different servers. So after the validation is over, similarly, we have to identify the active site of the protein, and then we have to add hydrogen charges for further docking analysis. So these are the few databases that we are using for the target preparation. First one is like PDB that I already informed. If the structure is not available in the PDB, you can go to UniProt database. From there, you can search your target. And if you are fortunate enough, nowadays AlphaFold is directly in AlphaFold in collaboration with UniProt. They are developing the structures and they are depositing with the UniProt. From there also, you can download the structure of the protein. Okay. If the structure is not available in both the databases, UniProt and PDB, then we have to perform either homology. We have to perform homology modeling, either using modeler, Swiss model, or you can go for AlphaFold. Or apart from that, if homology modeling is not possible for a particular protein, we should go for ab initio structure modeling, where also we can use AlphaFold, ITESR, and TR Rosetta. By using these types of tools and databases, we can get the target for a particular disease or a particular biological process for our further analysis. So after having that, I told you like high throughput virtual screening, we have reduced the number of molecules from the molecular database from 1 lakh to a few hundred. Then we can go for the dock-based virtual screening, docking-based virtual screening, where we have the target available with us. We have few hundreds of molecules with us. Now we go for virtual dock-based virtual screening, and from there we can end up into few molecules that are having high affinity for that particular target. High affinity for that particular, that is very important. The molecules, when they bind onto the target, disease target, that should be reversible. That should have high affinity, that should bind very effectively. But one thing we must remember, that the drug should not be irreversible. If, for example, a drug is creating a side effect and we have developed an irreversible drug, like many studies I have seen, people are doing covalent docking, that gives irreversible interaction. So covalent docking people are doing in order to destroy a particular target. For example, I am going to destroy a toxin. So in that case, it is fine. Irreversible docking is good. But whenever we are treating something in our body, preferably we should not go for the irreversible drugs, so that may create side effect. So in this kind of docking, the molecules go and bind with high affinity with the protein, and they are reversible type of interaction. Okay. Then once we are landed up with few 1 or 2 molecules from total 1 lakh of molecules that is having affinity for our drug target, we should optimize the structure of the molecule properly. That is called as geometry optimization. Most of us, we do not perform. What do? We get high throughput screening. We simply go for redocking. That means we have to validate the docking. For that, we'll do with the standard molecule. We do the redocking and go for molecular dynamics. But that is not the correct way. When we are downloading a molecule from a database, it is not properly arranged with their spaces in their geometry, with their charge, with their hybridized state. See, in order to determine that, in order to see the property of a molecule, whether they are reactive, non-reactive, okay, whether the geometrically the molecule is stable or not, we should perform the density functional theory. But due to the limitation of the resource, DFT tools are costly, like the Gaussian, many people they cannot buy, but many institutes they are having centrally, it is available. So from there people can take help, and by using the density functional theory, we can calculate the frontier orbital analysis. We can understand the reactivity of the molecule, whether the molecule is reactive or a nascent molecule, whether this molecule can be used for our further study or not. Okay, so this DFT will help us to determine the molecular property and the optimized geometry of the molecule. So once it is done, then our next step will be the redocking for validation. So in this case, we have the target with us. Take this geometrically optimized molecule, take this, and finally, what we have to do, we have to dock at the binding site, and we should also check that docking with the standard drug that is available in the market, or that may be available in the PDB structure when we have downloaded the protein. When we have downloaded the protein from PDB structure, we can see some molecules are already attached. That is called as co-crystallized ligand. That co-crystallized ligand, again, we have to redock at the same place, whether at the same place the docking is happening or not. In order to understand that, we are doing that. Okay, so if it is happening, then we can use our molecule to dock at that particular place to validate the process of our docking, whether the docking is fine or not. Apart from that, we can validate the docking by using decoy sets of molecules. So that is a different approach. So for that, we are making the ROC AUC curve to understand to better understand the molecular docking and their validation. Okay. When the docking is performed, so we have a complex with us. That means we have a target protein, target DNA, or a target RNA molecule with us, and a particular molecule that we are looking for, that is in the bound state with the protein, DNA, or the RNA. So, but how long will it bind? How effectively will it bind? When we perform docking, usually we do not consider water. We do not consider pressure. We do not consider temperature. We are performing docking in vacuum. That means in vacuum environment. So when there is no physiological environment available, then just relying on the docking is not enough. We have to create a virtual physiological environment where we have to perform, we have to understand the complex, the dock complex is stabilized in that particular physiological environment or not. For that, we are performing molecular dynamic simulation. So here I showed you a few of our research work that has been been published. Like we calculate root mean square deviation from the molecular dynamics, that is for 150 nanosecond dynamics. That means 150 nanosecond is very less. I'm telling you, for molecular docking, for drug and protein interaction, usually as per the molecular dynamics. Molecular dynamics says that if we are doing a interaction analysis between a target and a small molecule, between a target and a peptide, or a protein, there should be at least minimum 200 nanosecond interaction analysis we should study. Otherwise, interaction study below that point actually gives some vague information. Okay, so mostly, due to the computational resource, we are going for 100 nanosecond. Now I have a computational resource available. We are using 500 nanosecond, 1000 nanosecond. That is available with us. So we are working. Okay. So by doing this, this is about 150 nanosecond. This is published actually earlier, a few years back. So we have studied, and we can see we got a very stable RMSD. Based on the RMSD, I can tell the complex is a little bit complex, the protein ligand complex, or the RNA ligand complex or the target ligand complex is stable over the period of certain over the period of certain time. Okay, then we can calculate few other parameters like RMSF, root mean square fluctuation, radius of gyration, hydrogen bonding. We can go for free energy landscape. We want to see when the drug molecule binds with the protein or the macromolecule like DNA or RNA or a particular target, how the protein will behave, how whether the protein is distorted, whether the protein is refolded, what is the pattern of the fold of the protein, what is the energy of the protein. To determine that, we can perform the free energy landscape analysis. Then we can perform PCA to understand the principal components. We can say the efficiency of components in globular or local to maintain a global and local motion of a protein during the process of randomized simulation. Okay, then we can perform the cluster analysis to see, this is RMSD cluster. We can perform RMSD cluster analysis to see how many numbers of RMSDs they are below 3 angstrom. When they are below 3 angstrom, that has been considered that as a stable converse structure. Okay, that means protein ligand complex that we docked, that is a stable converse structure within a physiological environment. Then this is a picture of finally how we have performed the cluster analysis. From there, that has been developed. So here I would like to show a few of my publications that has been published in last few years. So here we have published in carbohydrate research. So these are few collaborative work. As I told you, open collaboration is very important from drug discovery. Otherwise, it is very difficult. So we have published in carbohydrate research. Then we have published in Journal of Molecular Journal of Medical Virology, Wiley. Then we have published in Environmental Science Pollution Research, European Journal of Pharmacology. Then few others like the JCMM, Journal of Cellular and Molecular Medicine. Then JBSD, it is well known to everyone. Then in Bentham, International Journal of Biological Macromolecules. So these are our few publications that we published in past few years based on the methods that I have mentioned. That network pharmacology, all those recently also we are going to we are already incorporated in our research, and we are going to get a good publication from there very soon. So this is the overview that I have given in my lecture. Identify the DEGs is very important, like differential express genes, then the Venn analysis, network and functional analysis, then drug discovery and validation process, then the ligand refinement and molecular interaction analysis. So these are the few methodologies that we can use for a research work, to carry out a research work. Here I have not included one thing which is called as QSAR, as because I have seen in the in your program that yesterday already Professor Masant has already given a comprehensive idea about the QSAR. So that's why I have not incorporated in my lecture. So by using these methodologies, these few steps, people can go for development of to understand or develop of a new lead compound, which can be a probable drug in future. After doing this kind of work, we can go for the synthesis work in the laboratory. Then we can go for the in vitro and in vivo analysis, then go for clinical phase trial analysis. Okay, so this way we can develop a new or a modern drug for a certain disease. We can also develop personalized medicines, many more things. So by saying this is my last slide, so this is my conclusion. So here I would like to attend some questions if you have in your mind. Thank you very much.
Thank you, sir. Thank you so much.
Hopefully my voice was audible.
Yes, sir.
Yeah.
It was very loud and clear. Thank you so
Yeah,
much.
I'm always loud. I don't need mic. Thank
The
you.
session was truly brainstorming, sir. Now the room is open for question and answer session. I request all the delegates to raise their hand if they have any question. If delegates are interested, they can also drop their in the chat box.
So I have a question. I can see one question. Is there any software to perform DFT? I told you, there are
Sir,
yeah, there are several softwares available. Like you can use Gaussian. Gaussian is one software, but that is that is a proprietary software. You have to purchase that. Or if you don't want to go for a Gaussian, you can use MOPAC, MOPAC. MOPAC you can use. Then there are other softwares like you can also use Schrodinger. In Schrodinger also you can use. That is not also free. The free softwares you can use MOPAC, and there is another tool which is called as that is I forgot the name of the tool. I have to see. So that is also free. But the problem with the free software, sir, there are limitations. You can use the open source software, but there are limitations. You cannot. The diagrams I showed you. So these are from Gaussian for DFT analysis. So that is in collaboration with the Jadavpur University we have performed. As because I do not have the license for that. So one of the faculty from Jadavpur University helped me. And I really believe on the open collaborative research. So we have performed our work accordingly. So let's see what is the next question. Can we do molecular simulation?
जलसा करे हाथ.
Yes. So Nikita Jalsakre, yes. So sir, now binding database is not working. So which software should binding? Okay, binding DB. Binding DB is not working, then you can use Campbell, right? You can go for Campbell, but in Campbell, yeah, from there also you can get the KI values. CHEMBL. Campbell you can use. But the binding DB is good. Binding DB is good, definitely. So Dhanashri, Garud, right? So
Yes.
can we do molecular simulation study for combination of drug, or we should go one by one? We can use for combination. Like virtual screening, the way we do, we cannot do. We have to go for one by one. But if you do combinatorial studies, like if you perform a combinatorial docking, okay, that means two drug together within a target, we can go for molecular dynamic simulation. You can perform. That means number of molecules that has been attached within a target, you can perform molecular docking, molecular dynamics. But it depends the tool that you are going to use. I showed you a few three tools like in my slide, like Gromax, Ember, Desmond. Okay, so if the molecular properties are not fulfilled within the force field criteria, it may hamper your work. So it's better to go for one by one. If you have two drugs together, that can also be performed, but multiple drugs may create some issue.
Okay, sir. Do we have any more question? Okay, sir. Now I request Chairman of the session, Dr. Sunil Jalalpure, sir, to conclude the session.
Yeah, thank you, madam, and very good afternoon. And thank you, Dr. Arabinda Ghosh, for your
Namaskar, sir. Namaskar.
long and very informative talk. Few slides of your presentation initial I missed because of some other work. Otherwise, I was there throughout your presentation. So nicely you have covered about how best in silico methods we can utilize for the drug discovery. So earlier these things were not available, so it used to take a long time for the drug discovery, and also need to spend a lot of money on drug discovery. So now time as well as money has been reduced by using these in silico methods. So you have also touched upon about artificial intelligence, then machine learning, how to develop the personalized medicine. Though these things are very important for our students as well as faculty members to understand about these things. And also you told very informatively about how the target identification can be done, then ligand selection criteria. So these were all in all need of the hour for the students to understand about these things. And one important thing you touched upon is different tools used in network pharmacology, especially the network visualization with cytoscope, then functional enrichment analysis. So these were told in very meticulously and in a lucid way to understand the delegates. So one important thing I liked is about the importance of finance to do the research activities. Nowadays, it is very difficult to carry out research activities without a financial assistance. So we will have to have sufficient amount of fund to do such kind of research activities, to procurement of equipment, chemicals, spend on manpower. So it requires money. So even one way is to do such type of research is collaborative research. If you don't have enough fund, money, so you join hands with others. So this I liked during your presentation you were mentioning about. So all in all, I think I thank Modern College of Pharmacy leader, visionary leader, Dr. Praveen Chaudhary, for arranging such a wonderful international conference and calling a very resourceful person, Dr. Arabinda Ghosh. And definitely it has benefited to all the delegates who all are attending this conference. And I wholeheartedly thank all the organizers for making me on this day as a chairperson for this session. And it was a wonderful session by Dr. Arabinda Ghosh. I congratulate and thank him for giving a good talk on the in silico methodologies in drug discovery. So thank you one and all, and thanks for the making me as a chairperson, and thanks Dr. Ghosh for your wonderful talk. And also I thank all the
Thank you,
delegates.
sir.
Around 150 odd delegates are attending online and sparing their time and listening to Dr. Ghosh. I thank them also. Thank you one and all.
Thank you so much, sir. We would also like to listen from co-chairman of the session, Dr. P. Parshuraman, sir.
Hello, is it audible?
Yes, sir.
Yes, thank you. So, good afternoon, one and all, and good afternoon, Arabinda, sir.
Good
Can
afternoon,
you
sir.
hear? Yes, sir. So, what are the points I want to share? Already, the chairperson.
Sorry to interrupt you. I request you to please keep your camera on.
Yes. Yes.
Thank you, sir.
Whatever the points I want to share before myself, our chairperson of the session, Jalapur Sunil sir, I have shared everything, like where you have started, how you have gone through. So usually the main thing in a drug discovery is target choosing, and how I can able to correlate that. You have and meticulously you have explained about the what do we call network pharmacology, and you touch all the domains, and you showed all the free tools, because max to max, as sir said, right. So financial point of view, so we can use such kind of tools, and we can able to publish. That you have proven at the last slide. Okay, with these tools I can able to publish in the best journals. So that's a wonderful thing, sir. And moreover, you touch all the domains as basic chemist or basic researchers or pharmacology, whomsoever it may be. If they touch to the work with the computational part, yes, enormously they might have got a complete throw into them. So that if they start to dig out and work on more, so far better, sir. So thank you for your wonderful lecture, sir. Happy to hear that.
Thank you.
Thank you, sir. So I would like to thank the organizing committee, Praveen Chaudhary sir and Sondata Chaudhary sir and Modern College of Pharmacy for giving me an opportunity to be a co-chairperson for this event. Thank you, one and all.
Thank you so much, sir. Now I request Speaker of the session, Dr. Arabinda Ghosh, to accept virtual felicitation with floral bouquet and certificate as a token of admiration and respect. Sir, kindly accept on behalf of convener and principal, Dr. P.D. Chaudhary, sir, Modern College of Pharmacy, Nigdi.
Thank you very much.
Kindly accept certificate as well, sir.
Yes, thank you very much for the certificate and the felicitation. Thank you very much.
I also request Chairman of the session, Dr. Sunil Jalalpure, sir, to accept virtual felicitation with floral bouquet and certificate as a token of admiration and respect.
Thank you. Thank you so much, madam, for the felicitation. Thank you.
I request co-chairman of the session, Dr. P. Parshuraman, to accept virtual felicitation with floral bouquet and certificate as a token of admiration and respect.
Thank you everyone.
Thank you so much. I hereby declare that scientific session has been concluded. I request all the delegates to fill feedback forms through a link shared on conference WhatsApp group. Your feedback is truly valuable for us. As we will be declaring results of poster and oral presentations, I request everyone to attend valedictory function and rejoin sharp at 3 p.m. through the same Zoom link. Thank you.